Everybody working live with Chaudhry here at PEGS Boston. I'm delighted to join by Roman Kurek from Samplix.
Yes.
And he's going to give me a quick overview of Samplix and what they took the delegates about here. So first of all, Roman, it's lovely to meet you.
Likewise.
How have the first 2 days of PEGS been for you?
It went amazingly well. So we had a lot of interesting contacts and conversations, a lot of leads already, and this is a pretty new area Samplix is looking into, so I'm very happy so far.
Brilliant, I'm glad to hear it. Now for those people who are not familiar with Samplix, can you give me a quick overview of the business?
Sure, I can. So Samplix is a Danish company, Copenhagen-based, founded 2012, and at that time the idea was to so there was the challenge to look into rare genomic variations in bacteria, and there was no technology out there to solve that. So the founders of the company decided to go 2 ways. One is to avoid PCR whenever possible, and secondly to work with droplets. So it took them a couple of years to come up with this instrument, which we call Xdrop. It's a droplet generator.
Right.
And it's a very flexible instrument because how it works is that it uses cartridges.
Right.
You can do up to 8 samples at a time. And the good thing is, as you can see, there are a couple of cartridges here, all doing different things. So the cartridge type actually decides what the instrument does.
Right, okay, and can you only use 1 cartridge at a time, or can you put more than 1 cartridge?
You can use only 1 cartridge at a time, but an additional flexibility is that you do not need to use the full cartridge, or you need to dump a cartridge when only have been using half of it. You can actually tell the instrument which lanes to use. You can store the cartridge for 2 to 3 weeks, and you can use the other 4 lanes another day. So that's another advantage of it.
Brilliant. And I see you've also got a screen there, so what is that showcasing?
Well, this is actually what we are now looking into, and that's why we are here at PEGS, is that we can encapsulate cells into droplets.
Right.
And it's important to notice that these are double-emulsion droplets. And that means it's water in oil in water. And that has a number of advantages. So these droplets are extremely stable.
Right.
You can PCR them, you can freeze them, you can ship them, you can store them, and you can sort them on a cell sorter. And that app actually opens up many potential applications you can do.
Right, so I'm not a scientist, so you said potential applications. So what are some of those potential applications?
Well, I don't want to go into detail where we started with. So we started actually putting DNA into droplets and developing a workflow for enriching long DNA fragments for long-read sequencing. But that's not why we are here. Or actually, we do have a poster over here. We can have a look later that shows a DNA application. But when it comes to here is that this is a flexible open platform. So customers coming to us asking, what can I do? And our answer is, what do you want to do? Because you can do many things, it's depending upon your assay, and we can help you getting it to work.
Fantastic. Now you mentioned you've got a couple of posters, so before we go to the posters, I'm going to pause this video, viewers, and by magic we'll be over at the posters, so bear with us 1 second.
Sure.
So hi everybody, we're now over at the first poster that Samplix have got. So Roman, would you like to talk me through your poster?
Yeah, so I don't want to go through all details, but this is an example for the DNA workflow we are doing, and it's shown here. It's a bit small, but what we actually do, we take DNA, large fragments of DNA, and we put them together with PCR consumables into our double-emulsion droplets we talked about. We use PCR to create a signal in the droplets that are of interest. And they show up in green compared to the droplets that are not of interest, and then we physically separate them by sorting them on a fax machine.
That's interesting.
That is interesting, right? Because and then we have to amplify those contents. So we break the emulsion and we amplify the containing DNA, And what it adds up is that we can actually sequence them on long-read sequencing instruments, Oxford Nanopore or Pacific Bioscience. And the beauty of it is that we can really recover and sequence long genomic regions. And this is of particular interest because this is an example for CAR-T insertion, so insertion of CAR-T cassettes in the genome. And when it comes to quality control, it is important to show, first of all, the insertion itself. Then you have to show that you don't mess up anything with the insertion, so creating mutations, SNPs, indels, whatsoever. And it's also of interest to show the number of cassettes that have been integrating into cells. And that's what you can do with this technology, because you can actually pick out the fragments containing the cassettes with the PCR assay specific for the CAR-T insert. But as we have long DNA fragments, you can look left and right. So you can identify the genomic location, you can identify potential mutations you have introduced. And this is just, I think, a very beautiful example showing an experiment with just 30 nanogram of DNA, which translates to 5,000 cells. And what you can see is roughly 1,000 CAR-T cassettes insertion sites. And what you can also see, there is no pattern, right? They were really randomly inserted in the human genome, so on each and every chromosome, even on the Y chromosome. And this is only possible with this technology.
Fantastic. Now, you have another poster, so
Yep.
viewers, I'm going to ask you just to bear with us for a split second,
Sure.
while we just go over to that as well. And we're back, and now we're over at the second poster we have here at PEGS. So, would you like to take me through this one, brother?
Yeah, so this is not the DNA workflow, this is the newly developed single cell resolution, so cellular workflow. So instead of putting DNA into our droplets, we put cells into droplets. And the idea is to go from a bulk assay with a mean outcome to a single cell assay with a specific outcome per cell. And these are just two examples we have been doing. So the idea is, this one is an interferon gamma assay that proves activation or no activation of cells. And the problem with, or the potential problem at least, with bulk assays is that you might have a crosstalk between activated and non-activated cells, and that might lead to a wrong output, so to say. And in droplets, you can actually put them separate, and you can keep them separate. So there is, during the incubation time, which by the way is shorter, and the signal is a bit brighter because these are very small volumes, you keep them separate. So you have no crosstalk between an activated and a non-activated cell.
Right.
And when you look at the outcome on cytometer, you can more clearly distinguish between the non-stimulated and the stimulated population compared to the bulk assay. So that's the first proof of concept. And the second proof of concept actually is to look for co-encapsulation of cells.
Right.
So here we have a co-encapsulation of a target cell and an effector cell, and we can stain them with different colors. So you can see the two cells here, and you can see the two signals here. And then we have just an assay for cell death, which is shown here. So this is a cell killing assay showing a potency of the effector cells. And as doing this in droplets, again, you don't get this cloud or, please excuse, messy signal you might get when you have a bulk assay where you have many different cells. You have activated cells, you have non-activated cells, you have effector cells, you have target cells. And when you look at this plot here, it's stained for the effector cell, no, for the target cell, and the dead stain signal. There are different populations of cells that are somehow in the same cloud. And when you do this in single cell, in single droplets, you get a much clearer discrimination between what's really going on. And this is a simple experiment showing the percentage of dead cells after 2 or 4 hours of incubation, including a control. So the whole idea is to go from bulk into an assay with single cell resolution.
Brilliant. Now, for people who are not here, where can they get more information about Samplix?
Well, you can get more information at our webpage at www.samplix.com, and you can always just click on contact us, and we are more than happy to get back to you.
Well, Roman, that's brilliant. Thank you for talking me through both of the posters as well. That was really interesting.
You're welcome.
So there you go, viewers. If you're here at PEGS this week, come over to the Samplix booth, talk to Roman and the team and find out more about what they're doing. And if you can't make it, then check out their webpage and also the LinkedIn page, and contact Roman directly if you want a link to them.
Sure, absolutely.
So Roman, thanks once again. It's lovely to see you.
You're welcome.
Viewers, thank you for watching. Until next time, stay well and stay safe. Bye-bye.